Hello, my name is Amie Eisfeld, and today I'm going to talk to you about microbiome analysis of urban and indigenous people in Colombia, South America. The funding for this work was provided by the Global Health Institute at the University of Wisconsin-Madison, and the work was carried out by a collaborative team at the National University of Colombia in Medellin and the University of Wisconsin-Madison, as well as the One Health Consortium. Human microbiota comprise all microorganisms that reside on or within human tissues and biofluids. Microbiota may have commensal, mutualistic, or detrimental relationships with human hosts, and many factors can affect the composition of the microbiome, including geography, ethnicity, and lifestyle. In addition, antibiotics are an extreme environmental pressure with possible long-term or irrevocable effects on healthy microbiome composition. Importantly, imbalances in a healthy microbiome are associated with human diseases, including infectious disease, and therefore understanding the properties of a healthy microbiome is essential for identifying and correcting microbial imbalances that may cause or exacerbate human disease. The goals of the current study were to compare microbiomes of individuals with and without extensive exposure to antibiotics, and to identify microbiota-associated risk factors of infectious disease or other human disease. To do this, we obtained microbiome specimens and epidemiological surveys of individuals in the city of Leticia, Amazonas, in Colombia, South America. Leticia is a major port of the Amazon River, and an indigenous community located 11 kilometers from Tarapaca Highway near Leticia, Amazonas. Here, it is important to note that indigenous communities typically have little to no exposure to Western antibiotics over the course of their lifetimes. The specimens that were collected for this study include nasal swabs, saliva, and fecal samples, which are to be used for microbiome analysis, as well as blood, which will be used for serological studies. We also collected epidemiological data, including demographics, medical history, vaccinations, socioeconomics, cultural aspects, and exposure to animals and insects. In our ongoing work, we are using serological assays to confirm infectious disease and vaccination history and work that's being carried out by our team in Colombia. And in addition, we are carrying out preliminary analysis of gastrointestinal microbiota by using fecal specimens, and work is being carried out by our US team. In future work, we will perform further analysis of microbiome specimens and microbiota populations, and we will also use statistical comparisons between microbiota populations and epidemiological data. Thank you for your attention.